Hi, dear friends from all over the world. Good morning, good afternoon, and good evening. I'm Chloe from RWD Life Science. Thank you for joining our webinar. The RWD Academy is a platform for researchers around the world to share their research achievements. In recent years, the use of fiber photometry to detect neural activity in the nervous system of awake, free-moving animals has become an important technique for studying emotion and behavioral neuroplasticity and neurodegenerative diseases. It's our honor to have Professor Francesco Papaleo and Professor Diego Scheggia as the webinar lecturer. Today, they will bring us a academy sharing about circuits of emotion recognition and social decision making from behavioral to neural mechanisms. Here is the schedule for the webinar. During the presentation, you are welcome to submit your questions in the chat box. Francisco and Diego will choose some questions to answer in the Q&A part. Especially if your questions are selected to answer, we'll give away our fiber optic news trial as a gift after the webinar. So don't hesitate to participate and ask questions. We look forward to your joining in the discussion. First, we invite Francisco to bring his report. Francisco focused on investigation of the mechanisms underlying social and cognitive processes that are altered in neurodevelopmental and psychiatric disorders, and used a cross-disciplinary approach including detailed studying in mouse and parallel clinical investigation in humans. His related research has been published in Nature Neuroscience, Molecular Neurodegeneration, among others. Today, he'll bring a presentation on the topic Circuits of Emotion Recognition, which includes illustrating behavioral paradigms to assess rodents' ability to detect and respond to emotional trends in others, and applying multifunctional techniques for understanding neural circuits underlying emotion recognition. Now let's get started. Hello, Francisco. Can you hear me? Please say hi to everyone and start your sharing.
Hello, hi. Yeah, maybe
Hi.
I will start to share the video. You should allow me for the video though.
Okay.
Okay. Here. Okay. Now you should see it.
Oh, now I can see your PowerPoint. Yes, it's okay.
Okay, so hello, thank you. Thank you again for inviting me and for giving us the opportunity to show you some of our work. So I will start then. Today I will talk about hello everybody. So today I will talk about our recent work on the circuits of emotion recognition. So in the last years, we've been focusing more and more on socio-cognitive processes, which by this we include a number of different abilities that are essential and fundamental for survival and for an efficient life in all mammals and all social species. Moreover, in our point of view, we study these evolutionarily conserved abilities also because for their importance on neurodevelopmental and psychiatric disorders for which we are interested. Indeed, common deficits in social cognition are characteristic for all these developmental and psychiatric disorders. Now, this has been studied a lot in humans, have been investigated in a different way, and social cognition have been addressed in different ways. But here is how we started to address in order to get some inputs on the mechanism underlying it. We started to divide them in three different layers, as I put you here. So starting from social perception, so the basic ability to detect and respond to altered emotion in others. This is usually studied by emotion recognition tasks. how we learn from others, so social learning paradigms, including observational learning, emotional contagion, and social anxiety, up to social decisions, so decisions that are made in social contexts and that can result in choices like altruistic or selfish, cooperation, and consolation. So as I said, this has been studied a lot in humans, but what was not studied instead was lacking in a lot of studies in preclinical studies in mice and rodents. So this is how we started to fill some gap in order to leverage all the potential that we have in this species to address mechanism with all the cutting edge techniques that we are available now. So today I will focus on this first layer, emotion recognition, and instead you will see then in the next talk by Diego Scheggia on his focus on instead of social decision making. So let's start from social for emotion recognition. What I mean by this, we started with this talking with psychiatrists how they measure this functioning. Usually they start to show to the subject, the testing subjects, phases of conspecifics, and the subjects need to discriminate what kind of emotions they are seeing. And this is, again, is very well studied, very well developed. But what we were doing at the time instead in mice was mostly checking how much a mouse was interacting with another without taking into account all this emotional component or this discriminatory behavior. This is why we started to think about, okay, could we maybe start to simulate or approximate key features of this kind of task in mice. This at the beginning was looking like a crazy idea, but was not so crazy. Indeed, here I summarize some of the work that we reviewed some years ago, in which there are a number of evidence from different animal species, mammals in this case, in which they have been reported indeed that is commonly able to show some expression of emotion, starting from sheep to horses, dogs, and primates. And I think who has dogs can understand this even more and better. Moreover, there are a number of different studies that have been flourishing in these last years in which we can dissect in different ways different social cognitive abilities, rodents, rats, and mice. And all of these studies indicate that indeed also rodents are sensitive to emotional states in others. So here I report some selection of them, the more refined paradigms, including this social modulation of pain, social buffering, helping behavior, consolatory behavior, observational learning, and so on. But at that point, none of these was indeed approximating the key features of this task that we were seeing. So this discriminatory behavior towards emotion and how this is usually performed in humans. And so for this reason, we came up with this simple test setting that we have been using and have been actually now validated and expanded in different ways by different labs around the world, providing this solidity and this flexibility in this usage. So what it is, I show you very briefly what it is. Then if you have questions, you can ask me more. So we have an observer mouse, which is our testing subjects, that is put in front of two demonstrators, and we will manipulate in different way and mildly the affective states of one of the two demonstrators in order to see how the observer reacts. So now I will show you some of the key manipulation that we are still doing, even though, as I told you, many more have been used and are used by other labs. So we first started with this manipulation that induce a negative affective states. So the idea was here to induce a negative affective states in one of the two demonstrators in the middle of the task, just to see how the observer will react to two demonstrators before that were in the basic state neutral, after the induction of unaltered states in one of them. So for this, we use a classical fear conditioning paradigm that was pairing a food shock with a tone 1 week or 1 day before the test, and then putting in front of the observer mouse this neutral, so not fear conditioned, and fear conditioned mouse before the presentation of the tone, 2 minutes during the presentation of the tone, and 2 minutes after. So what happened from the observer point of view? So what we saw was that for both males and females, indeed, immediately after the presentation of the tone, most of them, they were starting to prefer to spend more time investigating the fear mouse over the neutral. Importantly, I remind you, these two mice were neutral at the beginning, before the presentation of the tone. Here we were a little bit surprised to don't see a discriminatory behavior during the presentation of the tone, when indeed the fear mouse start to freeze because of the reaction to the tone. But we saw that this instead, indeed, seeing a mouse freezing was aversive for the observer. So only during the presentation of the tone, indeed, there is a anti-correlation to in which the observer mouse will avoid more a mouse that is freezing more. So this is the reason why you don't see this here. We then started to think about possible manipulation to induce a positive states, again, to simulate what is usually done in humans, in which you present positive and negative emotions. Here, we are still using these, despite have some limitations, but this is the one that is still most solid for us. The idea was to water deprive one of the two demonstrators for 1 day before the task, and then 1 hour immediately before the task, putting them back to full water. This is just to relieve them from a stressful situation, and we choose water specifically to don't give orders that could discriminate then the two demonstrators. We then checked that this indeed manipulation was inducing a positive affective states. Indeed, this manipulation induced a place preference and reduced corticosteroid levels. So we think that during this moment, the relief mouse is in a positive affective state. And what happened to the observer mouse when put in front of this neutral and relief mouse? Again, most of them, they were preferring to spend more time with the relief mouse over the neutral, even if they are males or females, especially in the first 2 minutes. And usually these kind of discriminatory behaviors tend to go away or diminish throughout the 6 minutes of the task. Another manipulation that we are using a lot is this mild stress manipulation. So it's again another negative manipulation in which one of the two demonstrators is restrained in a falcon tube for 50 minutes immediately before the task. And again, as you can see, especially in the first 2 minutes of the task, the mice will tend to explore more the stress mouse over the neutral. Now, until now, what we were doing was just manipulating the affective states of the demonstrators. However, from our own experience and actually from a lot of literature in humans, we know that our own experience will indeed influence how we react to others' emotions. And indeed, in humans, you can even find, for the same experience, opposite reactions. So some subjects might feel more other concerns, so what we sometimes call more empathic, so they will be more prone to be sensitive to altered emotional states in others and spend more time with them. Or you can have exactly the opposite phenotype. So some, they can remember that bad memory when seeing the other negative emotions in others, so feeling self-distress, and this will induce an avoidance or less sensitivity to other emotions. So in this case, what we thought to do was to see if this this could be evident even in mice. So Federica and others in the lab have been working on this. This paper was published recently. And what we did, so this is the naive mice, is the classical task, as I showed you before. The observer is not manipulated and is put in front of the restraint tube stress mouse and the neutral mouse. In the self-experience condition, the same stress was given to the observer mouse 1 day or even 1 week before the test. So what we saw in the observer, as you can see here on the left, as we always saw, for naive mice, most of them, they prefer to spend more time with the stress mouse over the neutral. However, what we started to see with the self-experience observer mice was a much bigger variability of the response. Some mice, they actually were loving more to stay with stress mouse over the neutral one. Instead, something that we usually don't see, some mice, they were starting to really avoid the stress mouse over the neutral. So this might have some similarities to what have been reported also in human studies. We then wanted to check here if indeed that stress manipulation that we are doing is impairing emotion discrimination in general. So we did some controls that were very important, in which we do the restrain, a Falcon tube restraint stress in the observer mouse and showing them the fear versus the neutral. But in this case, nothing was changing. They were exploring more the fear over the neutral as usual. We then wondered if we do the opposite. So we gave a foot shock to the observer mouse, but then put them in front of a restraint stress mouse and a neutral mouse. And even in this case, we didn't find any difference. So this really was suggesting that in order to see these differences, they should start to recognize the same exact stressful event that they were seeing before. So to prove this further, we did another protocol in which indeed the naive mice were exposed to a foot shock demonstrator and a neutral one. Instead, the observer was naive. And with the negative self-experience, we gave the same foot shock before to the observer and to one of the demonstrators. And again, as you can see here, while in naive mice, they prefer to spend more time with the foot shock mouse over the neutral one, the opposite is true for self-experience. So really, this was, I think, not trivial, because it means that despite not seeing the other one really stressed, they understand what's going on and they really understand that that was the specific stress through which they went before. So for this part that I just summarized here, our take-home message was that indeed we can start to see that mice can discriminate conspecifics based on emotional states. And we have been showing in different ways, again, also with other labs proving it, that this constitutes an additional tool to assess social cognition in mice, approximating key features of humans' emotional recognition tasks, and that check different social processes that compared to the other tasks that have been reported before. So now why we did all of this, as I said at the beginning, our goal was to take advantage of all the tools, technological tools that we have in mice in order to assess the neurobiology underlying such processes. And for us, this was really a Pandora box, because these processes were not investigated before. So we started in different directions. The first direction was checking for the oxytocin signaling. This because in humans, there are a lot of literature showing that oxytocin, especially, but mostly exogenous oxytocin, can influence emotional recognition abilities in humans. But when nothing was known at the level of endogenous oxytocin system. So indeed, the oxytocin mostly in the brain is produced by the paraventricular nucleus of the hypothalamus. We checked where these oxygenergic neurons are projecting to, and then by chemogenetics, we dissected one projection to another in order to see which one was more implicated in mediating emotional recognition abilities. And we saw that indeed the oxygenergic projection from the PVN to the central amygdala were the most involved in the ability to discriminate both negative and positive altered states in others. And this, I have to say, was already some nice indication that could bridge the world of preclinical studies with human studies, because amygdala is one of the major hubs that has been always involved in the response to altered emotions in others. So another big hub that is involved in emotional recognition in humans is the prefrontal cortex. So in this other set of experiments, we wondered if the prefrontal cortex in mice could also be implicated in emotional recognition. So we combined our simple test setting with the current available in vivo electrophysiological techniques just to record if the firing activity of the prefrontal cortex would respond to different emotions in others. We used optogenetics then in order to see if this pattern of activity could indeed be causative for their discriminatory behavior. And then we have been using miniscope techniques in order to differentiate if different subpopulation of neurons could be implicated in this discriminatory behavior. So combining all these techniques, what we found a few years ago now, that at the level of the prefrontal cortex, a subgroup of inhibitory neurons, these somatostatin positive cells, are very much more implicated in emotion discriminations. Instead, another subgroup of inhibitory neurons, like it has been shown by other colleagues in the past, are important, these PV cells, in social interaction when no emotions are involved. So based on this, we started to get more and more interested on these somatostatin neurons. So what they have, these neurons, that make them more and so particularly important for emotion discrimination. So in one set of experiments that also we published this year, we started to find that like was reported in the hippocampus, these somatostatin neurons are not the classical interneurons, but some of them, and in this case 10%, more or less like it was reported for the hippocampus, in the prefrontal cortex also 10% of them, they project long range in different brain regions. And we got more interesting and focused more on their projection to this brain region. Is called the retrosplenial cortex, which is in humans is embedded more in the posterior cingulate cortex. And why? We checked more this projection to these brain regions because together with the prefrontal cortex, this region is a major hub of the default mode network, which is implicated in psychiatric disorders for which we are interested, is one of the hubs that can be important for the so-called social brain network. And so we started to address if these specific inhibitory projections from the prefrontal cortex to the retrosplenial cortex could be important for mediating emotion recognition. So here I just put the summary of the data. Here we used a lot actually of fiber photometry techniques for imaging and recording and optogenetic techniques to check for the causality of this finding. But what we saw and what we found, what we demonstrated is that these somatostatin neurons projecting to the retrosplenial cortex, when they are in front of a emotional altered state, these induce an inhibition of this inhibitory input to the retrosplenial cortex. And this inhibition of an inhibitory input disinhibit pyramidal neurons that project back to the prefrontal cortex. And this inhibitory excitatory loop between the middle prefrontal cortex and the retrosplenial cortex is very important in showing the discriminatory behavior that these mice will show when put in front of an altered emotions compared to a neutral mouse. We started here to check also if this circuit could be indeed valuable or indicative for human. So in this case, this is was a study performed with the Schumann Lab in collaboration. Here they are still healthy subjects, so that we are around more than 1,000 healthy subjects that they go through a no emotion recognition, so exposed to angry faces, neutral faces, control, like an object or happy faces during a scanning. So the idea here is that, of course, with humans, we can reach a much lower mechanistic levels of precisions in what are the circuits and cell populations, but at the same time, we can have an entire view of the entire brain simultaneously. And so here, what I show you here is a summary of all the connection between the different hubs that we have been checking around the brain when presenting them an angry face, an happy face, a neutral, and a non face. And what I highlighted you here is indeed these blue dots here that are present only when the subjects are seeing an angry or happy faces. But these blue dots are not present when they see a neutral face and actually are red when they see an object. So these blue dots correspond to the connectivity between the prefrontal cortex and the retrosplenial cortex. And so blue dots means an anti-correlation. So this might make sense in the context of our inhibitory excitatory loop, with the idea that if one area gets active, the other one will deactivate and vice versa. So this will correspond possibly, probably, to an anti-correlation, which is only present when they see emotional alterations. So, but these, of course, will need more work to invalidate this and test this. So this was, let's say, for the basic emotional recognition. But as I told you, more recently also we have been checking how self-experience can impact on emotion recognition abilities. So here we started as well to check what would be the neurobiology underlying such processes. So here, of course, we first started to check somatosity neurons, but somatosity neurons we were seeing were equally important in emotion recognition with or without self-experience. So for self-experience, we wanted to check if something more specific was involved. And our first bet actually was to one of my first scientific loves, so was on the corticotropin releasing factor. This because the CRF system has been shown to modulate emotional and cognitive responses to stress. CRF is also produced and present in the prefrontal cortex, and CRF receptors are present in the prefrontal cortex. So our first question here was, what if we silence CRF only in the prefrontal cortex? Would this modulate emotion discrimination ability in our setting? So in order to do this, we first injected a virus that was silencing CRF only in the prefrontal cortex. And as you can see in naive mice, so observer mice that were not going through self-experience, this was not having effect. So it looks like CRF in the prefrontal cortex by itself has no impact on basic emotion discrimination. However, in self-experience mice, so mice that they had this self-experience, that overall, as I show you, some they prefer, some not, so overall they don't show any preference. When we silence CRF only in the prefrontal cortex, we start to see again this propensity to explore moral stress over the neutral mouse. So indicating that in this CRF, within the prefrontal cortex is important in modulating this self-experience, modulation of emotion recognition. So in order to check this better and physiologically, here we used the miniscope technique in order to image CRF neurons in the prefrontal cortex. Meanwhile, our mice were performing our emotion recognition task. Here, I think, was the most striking data. Because indeed, if you see in the naive mice, if anything, the CRF neurons are even inhibited when they interact with a stressed mouse, here in purple. But when they interact with a neutral mouse, actually there is some activation. But the opposite, a completely opposite phenotype is evident in self-experience mice. So you can notice here that when they interact with a stressed mouse, in this, there is an activation of CRF neurons in the prefrontal cortex, and there is no activation when they explore neutral mice. So we proved that this pattern of activity indeed could be important for the discriminatory behavior in different ways, in different optogenetic manipulations. So in order to do this, here I summarize you the data. We can go more into details later, but I wanted to summarize. Here you have in the first two lines the naive mice, here below the self-experience mice. In green, you have the relative activity of CRF neurons towards one of the two demonstrators, and below the corresponding preference for one of the two. So if you can notice, when we check and manipulate the CRF neurons in the prefrontal cortex in naive mice, it's kind of random effect. So sometimes you see that the one paired with a relative increased activation of CRF neurons is avoided, but sometimes it's preferred. Instead, we very much see a very consistent pattern of activity compared to the behavior when there is self-experience. So every time that the mouse is paired with self-experience with a relative increased activation of CRF neurons, this will induce an avoidance of that paired mouse. So again, this shows that these CRF neurons kind of store some sort of memory of that emotional states that is put online when they interact with that same emotions in others. And if this is activated, then they will show an avoidance behavior. So with this, I would like to conclude with the major findings, let's say, that we found so far, in which I show you that implication of oxytocinergic signaling in the central amygdala is important for emotion recognition. We started to address some prefrontal cortex-centered circuits subtending emotion recognition, in which we demonstrated that somatosity neurons has a special and central role. And we show that the self-experience modulatory effects on emotion recognition instead seems to be more selectively dependent on prefrontal cortex CRF neurons. So with this, I would like to thank all my amazing lab members, so current and past members, different collaborators from the preclinical side and from the clinical side, and different funding agencies that have been supporting us and allow us to perform this sort of test. And thank you for your attention. I'm ready for questions. Thank you. So at this point, sorry, where
Okay,
should I check the
oh,
questions?
okay, thanks for Francisco. Wonderful sharing. And Francisco left is currently hiring PhD fellow in emotional multi-brain dynamics, as well as postdocs. All interested researchers are welcome to contact him by email. Now it's time for the Q&A section. You are welcome to
Yes.
ask questions in the comment section. Francisco will choose the questions to answer for about 10 minutes. And the questioners will also have a chance to get the fiber optic cables as a gift. Now let's give the time completely to Francisco.
Yes, so I'm starting to check some questions. So rodents are night active animals. If you change the day-night cycle of the motion studies, have your screen also been performed? Yes. Okay. Yes, you're right. So most of our studies have been done during the day. So, but we have some preliminary data also that during the night phase, indeed, they show the same discriminatory behavior. And indeed, we did a long time ago a test in which we completely did the test in complete darkness. So in that case, if you remember for the fear versus neutral, in that case, they were preferring to explore more the fear mouse even during the presentation of the tone because they were not seeing the freezing behavior. So just to say that is possible, but this we will, I think, expand more on the night testing. Another questions. Yes, if why sometimes I choose chemogenetics and sometimes optogenetics. Yes, this depends on the questions. So sometimes at the beginning, for example, for the oxytocinergic project, we didn't have a clear answer or hypothesis on specific timing of the oxytocinergic signaling. So, and oxytocin also is released very slowly. So in that case, we wanted to give an inhibition of the system that was long-lasting. And this is reachable with chemogenetics. So it's and also chemogenetics has an advantage to be less invasive to the mice because they don't have any fiber. However, for other experiments, optogenetics give the possibility to manipulate the activity of the circuits in a very time-specific manner. So for those experiments that I show you that in the same test, we wanted to inhibit, for example, or excite neurons when interacting only with a mouse and not with the other in the same moment, in the same, let's say, period of time, optogenetics is superior. Okay, then may I know why 6 minutes? So the 6 minutes historically, let's say we choose them because as I show you as the fear manipulation, we wanted to see how they were responding before the presentation, when mice were neutral, so before the presentation of the tone, during the presentation of the tone, and after the presentation of the tone. So we choose these 2 minutes, 2 minutes, and 2 minutes. So this is historically the reason. Then with time, we saw that indeed the major discriminatory behavior, like I say, like I have to say, with like humans, is very fast. So even the first 2 minutes could be enough, but depends on your question. So most of the time, then we stick with the 6 minutes, but for other reason, for other projects, for example, we have been enlarging them, expanding to 50 minutes, so it depends. Another question. I'd like to know what are the miniscopes, how it is different from fiber photometry. Okay, and now you choose. Yes, this is a good question. Also this, we have been using both and depends on your question again. So, miniscope compared to the fiber photometry will show you single cell activity. So you will say but they are still, most of them, even there are some available that are 2 photon, but most of them are 1 photon. So you will see only the soma of the cell. Instead, fiber photometry, you will basically get an activity of the entire field of view, which also include dendrites, axon, etc. So depends on your question. So if you want to have information about the single neurons, at this point, of course, miniscopes are better. But for example, if you want to have information about fibers, so for example, terminals, fiber photometry in this case is superior. And also, of course, fiber photometry, it's easier and is also cheaper. So it depends on what is the question and if what of these two pros and cons you want to choose. So this is why we use all of them. And there are also some, for example, cell types that, for example, fiber photometry is still superior. For example, we have some projects on astrocytes, and fiber photometry is much better. Okay. Another question. Okay, so you speak of social and emotional settings as they are 2 distinct exclusive states. Does a mouse have sometimes emotional states regardless of the experimental induced? Okay, this depends. So the advantage compared to human studies, of course, that you can control what's going on with mice in the days before the testing. So we can really check that they were not undergoing any stressful situation. Then, of course, each one there an internal state that can change, but usually we take care that they are in a normal neutral states where they didn't have been exposed to strong positive or negative states. So this is why we can manipulate. But then, of course, the two things are very much linked. So this is why in the lab we made this link between emotions and social one of our center of investigation. So this is why we are so interested on them. Another questions here. Serotonin seems to be involved in emotional contagion in the lateral abenula. I know the work on Sarah and Mameli's group recently in Science. How do you think this parallel network interact with the oxytocin system? Okay, this is a good question. I don't know yet. So we have actually in the lab, Marconiello is bringing forward one set of experiments and projects checking the serotonergic involvement in emotion discrimination. So, and another postdoc in the lab, Ilaria Carta, is indeed working more on the oxytocinergic system again. So at certain point, we can try to link them and try to see if indeed this has some link. So we will tell you more in the future, I hope. Another questions. The mouse is more like an olfactory animal than a human as a visually animal. Do you think it can distinguish the negative or positive states of the anesthetic demonstrator based on their factor information? So yes, this is a good question. So we have been doing a lot of different tests in order to try to understand what are the sensory cues that drive their discriminatory behavior. And we found that it's a combination of olfactory and visual cues, despite, again, most people think that animals are not visual, but they are. So it's a combination of them. We did exclude auditory cues. We did exclude some tactile cues, even whiskers. The most important are visual and olfactory. But this, if you want to know more, because I would spend more time on this, please write me. You're free all to write me an email and I will tell you more. But yes, we have proof of that. Another questions, I'm wondering whether you have considered or looked at possible factory ultrasonic communication and how they shape. Yes, this, as I was saying, olfactory cues are very important. In our case, ultrasonic communication, no. But this also because we are using manipulations that are not inducing strong affective states, so they are not under pain. Okay, so we don't see any ultrasonic vocalization important. And also these are same-sex adult mice, and mice, they don't use a lot of ultrasonic vocalization in these settings. Another questions, I'm trying to respond to most of them. Let's see if I will be able. Is there a difference whether the demonstrator and observer mice are familiar or strangers? Yes, very good questions. Yes, there is a difference. So we see the same discriminatory behavior in familiars and non-familiar, but between familiars, we have a stronger discriminatory behavior, so which last longer, for example, for more than 6 minutes, and is stronger in nature. So it seems like knowing the other make them more tuned to them. And so this makes sense also for the human literature. Thank you. Another question. So what about the role of smell? All free mice in your task have olfactory information about each other, and it's likely influence their emotional states. Would you say that could be relevant for your results? As I said, yes. Odors is important. They follow the odor. How they transfer information between each other is something that we are studying much more now. And on most of the data that I show you, the responses were quick and faster. So most of them, indeed, most of the time there was not transfer information. Important also the demonstrators, they don't see each other and they smell relatively less each other. So also between them should be less important. But this is something that indeed now we have projects really focusing on all three of them simultaneously. Another questions. Did you see any difference in CRF concentration or any neuronal node activity in self-experience bias that show strong preference? So I don't know what you mean for, we didn't measure CRF levels because it's not doable. We tried with sensors, with fiber photometry, we didn't succeed. Was too shaky. Maybe in the prefrontal cortex there is not too much CRF, but we, as I show you, we silenced them or we checked how exciting or inhibiting them was influencing that. So this is what we know is involved in it. Okay, I saw eliminated the other question because I guess we reached the time. But I want to thanks again everybody that was connected, that have questions. If you still have questions, please write me. I will be happy to respond and happy to discuss for anything. And thanks again, RWD, for inviting me and for this occasion.
Okay, thank you, Professor Francesco Papaleo. Due to time limitation, this Q&A section will end here. And please take a break for Professor Papaleo. Before the next report, let's have a brief introduction to the RWD Life Science. RWD is a world-leading manufacturer of life science lab instruments and medical devices, specializing in the fields of screen and nursing, neuroscience research, animal behavior research, pathological analysis, and animal health. Neuroscience research requires good experimental instruments to help everyone with their experiments. RWD provides neural signal research solutions, including fiber photometry system, optogenetic system, stereotactic instrument, anesthesia machine, nanoliter injection pump, and micropipe puller are in hot trials now. Currently, RWD's fiber photometry system can be used in scenarios such as calcium ion and neurotransmitter signaling recording, neurocircuit research, neurological diseases research, and probe development detection. We have multi-channel fiber photometry combined with optogenetics and 4-color multi-channel fiber photometry system, and has the following advantage. One choreographs professional data analysis, high integration, and strong expansion. RWD's fiber photometry has worked in over 300 labs around the world. We have users from all over the world, such as MIT, Stanford University, Duke University, and so on. Welcome everyone to search our website and have applied. Now, welcome Diego to bring the next report. Diego focused on neural circuits governing social interaction and decision making, learning mechanisms in social behavior, and behavioral flexibility and adaptation in social contexts. Today, he brings the topic of frame circuits for social decision making, from behavioral to neural mechanisms. The main points of his reports include novel approach to understanding social decision making and novel mechanisms underlying social decisions. Let's begin. Hi, Diego. Can you hear me? Please greet everyone and start your presentation.
Hello, everyone. Yes, I can hear you very well. So I'm going to share my screen. I need to be allowed to share. If you can
Yes.
enable me. In the meantime, thanks a lot for your kind introduction and for inviting me. So just confirm if you can see my screen
Yes, it's clear.
presentation. Is it okay?
Yes.
Okay. So thanks everybody for joining this webinar. So I'm very happy that Francesco basically introduced everything you need to know for this presentation. Here we are going to focus on how we do select, let's say, appropriate behaviors in response to detection and recognition of emotionally relevant stimuli. So these behaviors and action that we take, that we are going to call social decisions, may have consequences on the others, such as in the case of other regarding behaviors that may benefit others, even at personal cost for the donor, or actions that are self-oriented and then that produce a benefit only for the decision maker. And these social decisions are very important because they contribute to shape inter-individual relationship. So as I said, Francesco introduced already everything. So let's say that the critical aspect of social decisions are the perception of so our ability to process social information that comes from the others, and also the possibility that our emotional mental state will match or not with the emotional state of the other subject in a social environment. It's something that we call in human neuroscience empathy. And then, of course, we use all these stimuli, and our brain will integrate these stimuli to respond with a behavior in a social environment. And why we are interested in all these aspects, as Francesco already said, some of these social cognition components are disrupted in many brain diseases, including neuropsychiatric and neurodegenerative disorders. So we decided to come up with a model of decision making in a social context using mice. Why we decided to use mice? Well, it's pretty evident after Francesco's presentation, but that is not the only ability that we have been detected in mice but in rodents in general in the last decades. We also know that both mice and rats can engage in behaviors that resemble human empathy. They can make helping behavior, so they can help their fellow. So in our aim was to develop a model to study choices that produce a benefit for the other at personal cost for the donor. Trying to create this animal model that could be used to study the underlying neurobiology, and then can be also used to eventually assess a dysfunctional aspect that may arise in specific brain circuits. So to start to develop this model of social decision making, we used a very famous human game called human dictator game, in which a dictator, which is the decision maker, decide to share or not a reward with the recipient. In the case of human, this is going to be a monetary reward, but of course, because mice do not care about money, we used food rewards. So we developed, we expanded the standard operand conditioning box with an additional compartment that hosts the recipient that basically may receive food rewards only upon the actor decisions. So the actor is presented, as you can see here in the picture, with two no spokes in which to deliver his response, his choice. One of the two no spoking will be associated with a shared reward between actor and recipient. That is what we label as we call pro-social choice. And the other one is associated with a self-oriented choice. So it's a selfish response that will deliver food reward only for the actor. So in the first experiment, we also use a control condition in which there's an actor, so the decision maker, without any social cue, so there's no recipient. And the recipient is a passive player, basically can only receive food reward. So we calculated a score to describe the responses of the decision maker, and we observed that most of the mice prefer to actually to share the food reward with the recipient. As you can see here, when you see the score as a positive value, that is indicate a pro-social choice, so it's a shared reward. While when it's negative, it's a selfish choice. You can see here our control animals performing chance level, so they close to zero. Not all the mice show a preference for pro-social choices. There are also many animals showing selfish preference. Then we also let's say we replicated this experiment many times. In the first case, we had a cohort of 52 animals, but now we have more than 300 animals, and mainly the distribution between the prosocial and selfish mice is more or less the same, so about 60 to 70% of the mice present this preference for sharing food resources. So to understand the nature of this behavior, of these choices, we decided to challenge the cost of these decisions. So we presented to the actor mice two options, modified in terms of cost. So the low cost option was the selfish choice. So one no spoke resulted in the delivery of one food reward. And the high cost choice was the prosocial one. So to make prosocial choices, to share food reward, the actor mice had to perform with the fixed ratio schedule that was increasing by 2. So 2 no spoke, then 4, then 6, and so on. And we found that basically only male mice accepted this extra effort in order to share food reward, while female mice and our, of course, control animals rapidly switched to the low-cost option. Then we also decided to challenge their motivation to make pro-social choices. And in this case, we tested mice for a longer session, like more than 2 hours. And we also modified the test in such a way that the pro-social choice was going to deliver food reward only to the recipient. So if the actor mouse wanted to have a food reward for himself, he had to make a selfish choice. And in this case, we found a very nice pattern of the separation of patterns between mice with mice showing a preference for prosocial or selfish choices, because the mice let's say the prosocial mice, first of all, they provided food reward for the recipient, and then they took a food reward for themselves later in the session. While the selfish mice, they basically did the opposite. So at the beginning, they provide all the food reward for themselves, and then they left some food reward. They made some prosocial choices, but only at the end of the test, basically when they were completely sated. That's very generous. So we investigated some of the social and non-social factors that could modulate this kind of behavior. We evaluated several of these, among them the food resources, availability, sex, familiarity, social hierarchy, and state matching, the ability of state matching. And we find that when considering these factors separately, let's say, as you can see here, the results were very heterogeneous. While by combining some of, even all of them, you can restrict, you can isolate a subgroup of animal showing, such as in this case, very high propensity for prosocial behavior. In this case, mice are food restricted, actor mice are food restricted, they're male, they have a familiar recipient, they're dominant over their recipient, and they also demonstrate very high emotional state matching. So then, we set up the model. Now we use the model to understand what happened in the brain when animals process this decision. So first of all, we focused our attention on the basolateral amygdala because we know that it was already known that it's involved in a number of social behavior, also in a number of decision-making paradigms, and we also have data showing an increased activation of the BLA using a CFOS, which is a marker of neural plasticity. So we decided to assess the causality of his role in decision making. We use a chemogenetic approach to downregulate, to silence the activity of the BLA during the social decision making task. And you can see that by silencing the BLA, mice lose this preference for sharing food reward. And also by analyzing individual learning curves, you can see that while control animal, they show a nice classical learning curve developing over time, BLA silence mice perform a bit at chance level. So probably also their learning phase was, learning abilities were disturbed by the silencing. So using fiber photometry, we wanted to understand in real time what was happening during their decisional processes. And we analyzed by using a calcium sensor in the BLA, in the principal neuron in the BLA. We analyzed their activity in response to the actor prosocial or selfish choices. Quite surprisingly, actually, I have to say, at the beginning, we found no big activation of the BLA, but then when the animals started to show a preference for prosocial or selfish choices, we see a higher activation of the BLA in the prosocial mice compared to the selfish mice. And also very interesting, this activation then disappeared when the task was fully acquired. So basically in the last days of the task. So then to assess the role of this activation in this smaller temporal window, we decided to come back to our chemogenetic experiment. And in this case, we decided to silence the BLA only in this specific time window. So in what we called a learning phase that corresponds to the second and third day of the task. And as you can see, that basically there's a drop of their preference in prosocial choices after this silencing. As you know, brain regions do not work in isolation, rather they work in concert with other partners in the brain. So to understand the brain circuit, we started to focus on the connection between the BLA and the prefrontal cortex by silencing neurons in the BLA, specifically projecting to the prefrontal cortex, which is one of the main input region from the BLA. By using a combination of virus, always using a chemogenetic approach, we basically replicated the same data. And we found that this preference of prosocial choices was disrupted, and also the learning was disturbed. So we also did the experiment by silencing neurons in the prefrontal cortex, projecting to the BLA by doing basically exactly the opposite, because the BLA is also a source of input from the prefrontal cortex. In particular, we focused on the prelimbic region of the prefrontal cortex. And in this case, while we didn't disrupt the learning at all, we basically we oriented the decisions of the actor mice towards the selfish choices. So by manipulating this projection, we started to elucidate the brain circuit of this social decision. So in one side, projection from the BLA to the prefrontal cortex guide the establishment of prosocial actions, while vice versa, prefrontal cortex to BLA projection exerts control over self-decision by using this task in which we could assess individual propensity for prosocial and selfish choices. And we also, using this task, we also assess some of the factors that might modulate these choices. So in the next part, I will focus on the learning, let's say, component of these choices, because we are very interested in understanding how much of these choices is innate or learned through experience or through previous experience. So far, I show you data of animals learning to make prosocial choices by self-experience, by repetitive trial and error. But as also Francesco mentioned, animals and also humans learn also by observing the others. Take, for example, this example. So you have one mouse observing another mouse, a conspecific, who is facing a stressful situation, in which he has to take action, make a decision, an imminent decision very quickly, because it's a decision, let's say it's for survival. There will be, first of all, a transfer of information from the demonstrator to the mouse who's observing. That will also match his emotional state, so he will present the same behavioral response, which is freezing, because it's a stressful situation. And this will also lead to learning. And this paradigm, which is widely used in the lab and under the name of observational fear learning or vicarious fear conditioning, has been used a lot recently to assess the neurobiology underlying this observational learning, which include also the amygdala, cortical region, and the hippocampus as well. But this paradigm has been used only in the context of aversive experience, so in the context of negative stimuli. So we were wondering whether an animal could actually learn in the same way other kind of information beyond fear. So we basically adapted our task to understand whether the recipient, that in this case act as an observer mouse, could learn from the decision of the demonstrator how to make a prosocial choice. So we exactly use the same setting. We added a second phase, which is a phase of decision making that follow the phase of observation. So basically, after 5 days of observation, the role of the observer and the decision maker are swapped, and the observer become the decision maker. First of all, we see that observer mice showed a higher number of choices compared to demonstrator. So they already know, already from the first day, so they already know the non-spoke and outcome association. And these choices were also more coherent. So we quantified their choices by constructing this index, which is a kind of preference index, tells you how coherent the responses were compared to the day before. And you can see that already from the first day, the choices of the observer were more coherent, suggesting that these mice already let's say, formed a preference before acting in the task. We also look at, let's say, a bigger population of animals. In this case, we have 56 animals, as you can see. By using this index, we calculated the number of animals showing higher or lower coherence index. We set a cutoff for this index to discriminate between animals showing a high preference or a low preference at the first day, and you can see the striking difference between demonstrator and observers, which then become similar only at the end of the task. Because it's a social task, they can display a social preference. We also analyze their actual preference. So here you can see also the distribution of the choices between demonstrator and observer. Again, here you can see how the choices of the observer are nicely distributed. This is the decision preference score, so positive value reflect prosocial preference, while negative reflect negative selfish preference. And you can see again here that while demonstrator perform a chance level at the beginning, already the first day mice, only a small proportion of observer mice do not show this a clear preference already at the beginning, let's say. So at the end of the decision-making phase, then we analyzed the data of the observer by dividing the observer based on their actual preference. So we divided observer mice based on their selfish or prosocial preference. And you can see that actually mice with a prosocial preference also show a higher number of choices, and also these choices were more coherent throughout the task. So suggesting that the individual prosocial propensity could be somehow facilitating the social transfer, the learning. So we decided to translate this preference into a more standard setting, which is the very famous and widely used three-chamber task. In this case, we presented the observer with their demonstrator or with an object. And in this case, you can see that while the prosocial observer show a preference for interaction, for exploration of their demonstrator, so of their recipient, a selfish observer, actually they do avoid their demonstrator, suggesting that their choices were goal-directed. Then to check to exclude that this was not a social impairment, we also changed the stimuli with novel stimuli with stranger mice, and we don't see this difference anymore. Then we wanted to explore the role of the model of the demonstrator that could impact on their on the social learning of the observer. In this case, we took all the data from the demonstrator of the observer, and we used a PCA that nicely discriminates the data by showing separation of data based on the preference, the prosocial and selfish preference of the mice, and also based on their role, on the observer and the demonstrator. We also look at the component that was more important that drove this separation, and we found that were the prosocial choices. So we plot these prosocial choices together, prosocial choices of the demonstrator. We look at the demonstrator prosocial choices, so at the model, and we plot it together with the coherence index, so how coherent were the responses of the observer. And you can see that these nicely correlated, suggesting that the higher the number of prosocial choices of the demonstrator, the better was the social learning of the observer. So far I showed you that mice can learn prosocial choices from observation of their conspecific. Observational learning is a component of prosocial behavior. And I also show you that the learning of prosocial behavior is enhanced by shared reward between demonstrator and the observer, and by the individual propensity for the observer. We then use this model to understand again what's the underlying neurobiology. We used again CFOS, which is a marker of synaptic plasticity, and interestingly, we found an higher activation of the dorsal CA1 compared to the other regions in the observer, but also compared to the dorsal CA1 of the demonstrator. So we decided to, we also looked at how this increased activation of CFOS impacted on the structural morphology of dendritic spines, which is of course, the place where synaptic plasticity happens. So this increase of CFOS also is reflected in modification of mature spine, which is the mushroom spine in observer compared to the demonstrator. So this create, let's say, a long-term effect. So again, to determine causal relationship, we used chemogenetics. In this case, we silenced the dorsolateral, the dorsal CA1, and as control also the ventral CA1, because the hippocampus extend from dorsal to ventral. And we silenced the dorsal and ventral CA1 during the observation phase. So then observer mice were tested without any silencing. And as you can see, while the silencing of the dorsal CA1 impaired the social learning, we observed no effect in the silencing of the ventral CA1. We look at the neural activity by using a fiber photometry, also in this case. So we analyze the neural activity of the dorsal C1 of observer mice during the observation phase. Then we tested them in the decision-making, and we analyzed the data considering their preference for prosocial or selfish choices. And interestingly, we find that prosocial observer, they show a striking drop of neural activity when they observe their demonstrator making sharing choices, so giving food rewards to both of them, while selfish observer, they show a completely different pattern. They show a rapid and short activation of neurons in the dorsal C1. So to understand if these different neural patterns are functional, if they mean something, we ask whether we could artificially recreate these neuronal patterns in the dorsal CA1. And in this case, we did it using optogenetics. So we use an inhibitory optogenetics to recreate this drop in neural activity. And at the end of the observation, we test mice in the decision-making phase, and we found that all of them show a preference for prosocial choices. And similarly, we recreated the neural pattern by producing a short activation of these neurons. And in this case, we oriented mice choices toward more selfish options. And this, you can see how differ compared to our control mice that more or less show the same distribution between as I showed you before. So by recreating artificially this neural pattern in the dorsal C1, we oriented mice preference by manipulating only their learning phase, not their decision-making component. To conclude, I also show you that the dorsal CA1 is involved in this circuit, and this circuit we are trying to depict, and that underlying social decision making, considering also how this behavioral repertoire are acquired through self-experience or through observation of the other. There's still a long run. We are just at the beginning. So for the moment, my take-home message is just that to be altruistic, a big heart is not enough. You need a brain. And I want to just conclude by saying thank you to all the members of our big research group, including Monica Di Luca, Fabrizio Cardoni, and Elena Marcello, all the guys who produced most of the data, mainly Filippo La Greca, Ilaria Cerchiaro, Michela Gusmini, and Alexander Carrea, and of course all the other members that contributed to this project. And I want to say thank you for your attention, and I will be more than happy to take questions if there are any.
Okay, thanks to Diego for the excellent report. Now is for the Q&A part. He will choose some questions from the audience to answer for about 10 minutes. Now let's give the time to Diego.
So here are the questions. The learned decision-making by observation model used in your research reminds me of the social threat safety test. Are you the first to use this observer decision learning behavioral model? No, I'm not the first, although there are very few paradigms that have been used to assess observational learning in contexts that are different to let's say, threatful conditions, such as observational fear conditioning. There are other paradigms in which animals learn to make action. These are more motor learning. Well, in our work, we show that this is not just a mere effect of mimicry, so they don't just imitate their model. But here I didn't have the time to show you all the data, but they learn to make a goal-directed behavior. I see that you have done gender correlation and disparity studies. Have you done age correlation? Age correlation, yes. We are now testing, so we have been testing mice of, let's say, across of the lifespan from adult mice So today I show you data from adult mice. From young adult, usually we used 3 months old mice up to 12 to 15 months old mice. And we see that actually there is a tendency to this prosocial choice, this prosocial preference tend to disappear over time. What we don't know is if this prosocial propensity is also present before the adult age. So we are now testing adolescent mice. Have you specifically tested for certain type of neurotransmitter in addition to GCAMP, like the dopamine reward system? Yeah, no, that's a very good point. We did not, but when we'll start from dopamine. I'm very interested in the reward system in this paradigm. So this is a long question. It looks like it's going to be a difficult one. So let's see. Previous observers that received more prosocial actions before were also more prosocial themselves as decision makers and more coherent in their decision early on, correct? So would you say this is just due to social facilitation in learning, due to more focused attention? Because when the decision maker is prosocial, the decision actually matters to the observer. You're right. Is there a tit for tat or a generalized reciprocity logic behind? So I think that yeah, you're right. It's probably that the prosocial observer are better in what they're doing because, of course, they received um, more reward, so they shared more reward, and of course, this has a higher valence, no, compared to nothing, compared to just looking at the other taking food reward for himself. So this could be a possibility. And I think that it's also related to reciprocity. Indeed, what I didn't show you is that if you change the partners, so if you, when the observer learn from a partner, but then you test him with a different one, you don't see this striking increase in choices compared to the demonstrator anymore. Probably because it's related to so they learn to also to reciprocate their so it's probably related to this aspect. Are all the prosocial or selfish choice measurements conducted between same-sex mice? Yes, these are always sex-matched animal. If not, considering mating desire, were there any neurobiological differences? We don't know, but yeah, the problem is that then this is going to be extremely different because, of course, sex will probably matter more than the food reward, and this is going to be like more sexual, social sexual interaction. So I'm pretty sure it's going to be very different. About the neurobiological differences, I have no data to answer, but this is a very important question. But there are so many aspects you could look at that we needed to select at the beginning. I was wondering, how do you distinguish whether the observer or observer mice learned the prosocial behavior by observing, or the observer mice was just mimicking no spoking in order to gain more reward where they acted as demonstrator. Yes, what I didn't show is that there is some flexibility in their behavior. So first of all, if you change the location of if you change the, let's say, the association of the no spoke with the prosocial of selfish choices um, between demonstrator and observer, they, I mean, you don't see a bias in the location of these choices. You don't see a bias also in the location of the left and right no spokes if they are closer. So they're close or distant compared to the observer. And as I said before, also if you change the partner, you see a modulation of their behavior. So there is some sort of flexibility. So it's not just pure imitation. Can you comment more on the selfish behavior shown by female mice? Yeah, so I think that so at that time we just said we label them as selfish because that's how we measure responses. I don't think that they are selfish by nature. So probably they're just more conservative. And this could be due to, let's say, biological reasons, as they maybe it's something innate, as you could imagine. Females, they have maternal, let's say, impulses that males they don't have. So probably they're just more conservative, and they're probably making choices in this way just because they are thinking about themselves first for these reasons, but not because they are more selfish in general. I think do you know that there's this last question? Do you know whether there are sex differences depending on the age of the observer mouse? No, I don't know. So we know that the age is a factor, but I don't know whether this will change depending on the sex. I will have a look at it. Thank you. This is a good point.
Okay, thank you, Diego, for the excellent report and answers. Now, more than 8,000 articles have been published using fiber photometry, and it has become a necessary technology for neuroscience research. RWD's fiber photometry system can be applied for a free trial. If you would like to get more detailed information and try the instrument yourself, we can provide an on-site demo. Welcome to contact us. For those who are selected by Francesco and Diego to answer questions, we'll contact you after webinar to confirm the information of the fiber optic canals delivery. Once again, thanks Professor Francesco Papaleo and Professor Diego schedule wonderful course. If you still have any questions, welcome to contact Francesco and Diego by email. We also welcome you to cooperate with Professor Francisco and Professor Diego Laboratory and RWD Lab Science. Finally, thanks everyone joining our webinar. Thank you, Professor Francisco. Thank you, Professor Diego. Thank you, everyone. Wish you have a nice day. See you next time. Bye. Francisco and Diego.
Bye
Ciao,
bye.
ciao.
Bye
Bye
bye.
bye.
Thank you. Bye.
Thank you.